My lab is a molecular biology lab that researches CRISPR biology and DNA repair. I've been working on CRISPR systems since 2002, since before they were called CRISPR systems, simply because originally CRISPR systems were thought to be DNA repair systems, which turned out to be an incorrect hypothesis. We all know they're now adaptive immune systems. However, we've carried on working on that, and so I've built some expertise in my lab working on CRISPR systems and how they work with DNA repair. The reason I'm sitting here now talking to you and hopefully listening to what you've got to say shortly is because through the University of Nottingham organisation that liaises between Nottingham and other universities around the world, I noticed an email where I think you and your team were looking to detect using PCR DNA signatures, etc., from Mycobacterium tuberculosis, I think. And in the past, I've been interested in teaching and researching using alternatives to PCR for this kind of detection, things that are sometimes called the Sherlock or the detector systems using Cas13 and Cas12 CRISPR-Cas enzymes. So that is my background, and I'm happy to listen to what you have to say in your proposal and make any comments, and then we'll go from there.
Great, thanks, Ed, for your introduction. So, because here we have a lot of teams, so just quick summarize. So basically, we have two majors in discipline. So some of us are in medical, some doctors and physicians, and the other is working on IT, especially on big data, data science, and AI. So we came from the multidisciplinary field of science. So, and at the moment, we have been shortlisted by the Indonesian Ministry Government to control a proposal. So this opportunity, we would like to present some of our initial idea, and we will happy to hear from your comment to improve our proposal. And anything that would you like to comment or make suggestion, it would be great. So anyone from our group will present to add, please. Okay.
Good morning, Professor Ed, and good afternoon, esteemed colleagues. Today, I'm excited to present our proposal titled Unlocking Insight, Development of a DNA Database from Tuberculosis Patients in Indonesia using PCR, as a part of our UKRI grant. I am Badar Katamsi, and I'll be walking you through our proposal today. Let's delve into our ambitious project aimed at combating tuberculosis in Indonesia. Before we started, allow me to introduce our team member who have been instrumental in shaping this project. Our team made up of 4 professors in UPN and Trunojoyo University. For first is Mr. Wahyudi and Dr. Gede, Dr. Rusdi, Dr. Yana, and for the last, Dr. Sharif. And 8 research student in UPN. First, me, Badar, and Fadli, Toting, Rangga, Pangestu, Elvira, Revi, and Davania. Our UKRI grant aims to develop a comprehensive DNA database from tuberculosis patients in Indonesia using PCR technology. This initiative holds promise for enhancing our understanding of TB epidemiology and creating target intervention. For explanation about TB cases in Indonesia, let me introduce you to Dr. Rusdi.
Okay, perfect. Good morning. I will do this. Myself, represented in TB, yeah, tuberculosis or TB is determined significantly how wounded. Yeah. Within the global Indonesia, especially, we have very particularly challenge, and this is a complicated case, yeah, in Indonesia, because TB is common disease in Indonesia. So despite effort to control TB, this is very important thing, because TB is the most complicated with the diagnosis, because the diagnosis of the TB is many have many things here. As you know, in Indonesia, we have two ways to diagnose TB. So, first is with the radiology, especially the thorax photo, and the second is with the photo culture. This is the why we need to learn the TB as well. Okay. The next is this is okay. Stop. Okay. In 2020, okay. Okay. The COVID-19 is very particular, because this is the state and of the TB and the diagnosis. Yeah. This way. Sorry, my connection is oh, sorry. Okay. The fact in Indonesia, yeah, the fact in Indonesia has the second highest number TB in the case. It is the world in the world, as you can see. And okay, next. The next 5 of the 10 individuals affected with the tuberculosis with no recovery in the 2012 and the 2019 after undergoing treatment. And the lowest number of the recoveries was only 2019 individuals, and the highest was the 1,407 individuals of the individual affected with the tuberculosis. Okay. Next. Okay. This is the database of the TB. Sorry. What the number slide? Okay. Sorry.
Sorry, Dr. Rusdi, I couldn't hear your voice. Could you repeat, please?
What the number of this slide?
Oh, this slide is already on 7. This is
Okay.
actually the Dr. Riana's.
Okay. Dr. Diana.
Dr. Riana, could you hear my voice?
Okay.
Okay.
Thanks to the opportunity. I'm Riana. I'm a clinical pathology. May I continue the presentation?
Yep, go ahead. I can hear you now clearly.
Comprehensive data on TB strain circulation within Indonesia hampers effective disease management and intervention strategies. Without a thorough understanding of TB epidemiology at the molecular level, healthcare authorities struggle to implement targeted measures to curb transmission and improve treatment outcomes. Next. Additionally, the emergence of drug-resistant TB strain further complicates the situation, posing a serious threat to public health. Without accurate and up-to-date information on TB genetic variation and transmission patterns, efforts to combat the disease remain limited in their effectiveness. Next. Hence, there's an urgent need for innovative approach to enhance our understanding of TB epidemiology in Indonesia by developing a DNA database from TB patterns using polymerase chain reaction or PCR technology. We aim to fill this crucial knowledge gap and pave the way for more effective TB control strategies. Next. Our UKRI, grant aim to develop a comprehensive DNA database from tuberculosis pattern in Indonesia using polymerase chain reaction technology. This initiative holds promise for enhancing our understanding of TB epidemiology and guiding targeting intervention. Next, I think the next is Mr. Pangestu. Please.
Can I just jump in here? I'm still on slide 7. Should we be on slide 8 now? Correct?
Yes, correct.
Yes, Prof. Ed. So actually, the Rihanna slides is only on 7 slides. So yeah,
That's good.
not on the
Thank you.
same slides. Yeah.
Good, thank you.
Okay, our project's key objective includes samples collection, DNA extraction, and PCR analysis, establishing a robust DNA database, data analysis, and collaboration with Indonesian healthcare authorities. And here's our aims. First, we will build the database pipeline in 2024 until 2025. This step includes extracting DNA data with the help from the local institution, including local government and NGOs. The second, we will building an AI model to approach a more efficient way to extracting a position of DNA that unique with TB. And then in the last aim, we have genetic marker, which we have not decided yet. Next. So the first year, our focus will be on developing comprehensive database pipeline. In the second year, we will make design for machine learning. Maybe we will use transfer learning and model aimed at predicting TB. In addition, if machine learning is not possible, creating user-friendly BioPython program. In the third year, identity-specific genetic markers that are significantly associated with TB disease risk with BioPython. So for the first aim, we will be discussing about that database DNA pipeline. We have Rangga in here to explain the DB of DNA.
Okay, thank you, Pangestu. In this database, the data that we're using is PCR result in the form of FASTA files. In this FASTA file, data will display in the form of genotypes that have been taken from the PCR process. The data stored in the database contains the ID for the genetic representation. And then we have genotype obtained from the FASTA files and SNPs for identify the FASTA data into a more structured form. Once the data has been identified, it will be put into specific categories that describe the location of each data on the human genome based on the genotype strand using the Human Assembly Reference Build 36. The data stored in the database contain RSIDs that represent unique identifier for single nucleotide polymorphism SNPs in the human genome. And in here we have chromosome entities to input chromosome values. And next we have position refers to the physical location of genes, SNPs are other genetic variants on chromosome, and genotype is the combination of the alleles and individual process for a specific gene. Data organization like this aims to make it easier to identify certain markers in the DNA that indicate the presence of TB. On the next slide, there is a use case diagram which will be explained by Dodin.
Okay, thank you for the time, Rangga. In this diagram, you can see we have two entities, which are admin and user. For admin has a broad range of capabilities, including managing the database, user management, and role management. In database management, admin have tasks such as inputting, selecting, and editing data in the database. For its roles as user management, the admin has tasks such as adding new roles for the user to be able to earn certain capabilities and database. Also, admin has authority to remove users' capabilities and access from database system. And for the users have more limited interaction, primarily focused on logging in, requesting access, and viewing or downloading data. And then for the M2, we will have to bio pattern that will be explained by Revi.
Thanks for the time. The models that we will use for this project is BioPython. There are advantages of using BioPython in this project, including facilitates the processing and analysis of biological data, including protein and DNA sequence, and then enhances efficiency by optimizing the import, process, and analysis of sequence data. And the last, BioPython's modules are designed for the tasks critical to viral DNA identification and classification, including sequence alignment, motif searching, and phylogenetic analysis.
For the conclusion, the project focused on building DNA database for tuberculosis patients in Indonesia by extracting genetic material using PCR. With Indonesia's high TB burden and limited genetic data, this research fills a critical gap, paving the way for targeted technological advancement in TB management and control. I extend my gratitude to Professor Ed and the audience for their attention. I'm now open to questions and further discussion.
Thank you. So you've got a big team for a big problem. The big problem being you're going to set up a database and then try to use that database from the start. Did I hear correctly that there's 1 million cases of TB in Indonesia every year, yet there's very little clinical database available? When you're gonna, I've got a couple of questions. The project now makes a great deal more sense to me than the very brief text that I was sent through by email from Nottingham. It was a nice presentation, thank you. I only lost sound once, but I don't think it matters too much because I now understand what you're trying to do, and it's great. The first question is a kind of ethical or database protection question. In the UK, in UKRI, there are very strict controls on patient information, the storage of data in your databases. So the question is, if you manage to produce this database from the beginning, from scratch, as I think you will, what do you have in mind to convince the funders in the UK, if it's a UKRI project, that the database will be carefully protected and all the ethical procedures will be put in place to do that? You'll need to be mindful of that if with anything in the UK. We've got very strict rules on storing patient data. I don't know if you want to respond to that, or I can carry on asking a couple more technical questions and give you some thoughts on where I think this might go regarding my knowledge of CRISPR systems.
Yes, please carry on and we will answer that later. Thank you, Prof.
At the end, yeah, okay, that's good. So my understanding now is that the first phase of this is to use AI and use algorithms and get the database up and running. I think that's very sensible, because that's the nuts and bolts of the project that provides the platform to then test it. But that's interconnected with using PCR to detect differences in the sequences that you're looking for. So you're going to be looking for different strains, presumably, of Mycobacterium tuberculosis, and determine which alleles are present is another question using PCR, which seems fine to me, although I'm interested to know specifically what you're going to target in Mycobacterium tuberculosis genome. Then I can see there's a direct opening to use the database and the PCR methodology to take it into the field. So at the moment, it strikes me as being a project whereby it will be very much centre focused on major city university clinics, research centres. I think that's another question. Once you've developed the database, that should give you the digital information, the DNA information that you require to be able to then take it cheaply and with much more sensitivity out of the clinic or out of the research centre into the field. Is that the plan for this, to build on AIM3, the genetic marker, and then make it something that you can use outside of the clinic? So you can, rather like Zika virus was analysed in South America by using mobile detection systems. I'm wondering what the plan is after you've built a database and collated all your PCR data. That's where I think the CRISPR angle might come in as the end point for highly mobile, highly localized, very cheap and highly sensitive detection of your TB markers. Okay, a few questions there. I don't know if you want to respond.
Okay, thanks, Ed, for your questions, and I'm trying to answer the first question. So regarding the sensitivity of the data of TB, and yes, we are collecting from the scratch from Indonesia. So I think the data is from Indonesia, so we would not getting the data from UK. And we also have been applied for ethical for the collecting the data. So all the data will be collected. We have, I think, if I'm not wrong, anyone can correct me. We have a partner hospital, yeah, in Indonesia, and then from there we have collaboration, and we're collecting the data from there. So it's not from UK. From UK side is a partner who will as I think as an expert, an expert, and the case is because the title of the UKRI is Southeast Asia infectious disease. So the project based the context in South Asia, and what we like to get from the UK is your expertise and probably some of the more advanced technique in detecting or marking something else. That would be great for us. So we have here in the contact and case study, and some of probably the modern on advanced technique from UK or your expertise will help us more make this research more advanced. I think the first question, and anyone can help answer the second and third question, please.
Okay, Professor Ed, could you hear my voice? It seems
I
fine.
can, loud and clear. Thank you.
Okay, so for the AI method, actually it's set on 20 and 25. So right now we are currently focusing on the database pipeline itself. So for the AI, we are currently developing the model. So for the sequencing and which position that we are actually trying to give to the AI, we are not fully decided yet, because the database itself it's on the working. So for answering that, we are currently still working on the model itself, and while trying to get more the data. That's all that we could say for now.
Okay, that's clear. Thank you. I'm most interested in, I don't have expertise in building databases, and I certainly don't have expertise in developing algorithms and using AI. That's way beyond my field of knowledge. There are people at Nottingham that do have that expertise, but I'm not one of them. I'm really interested in where you can take it, where you can go with this. And I'd be very happy to be involved as a named collaborator. I don't know the scheme that you're applying for through UKRI, but if they have named collaborators or expert consultants for how the technology could be then utilized, building on the PCR data to make this a CRISPR-based diagnostic. In the UK, I am quite well known in the CRISPR field. There aren't many CRISPR experts in the UK, but I would say I'm probably one of them. And consequently, I would be happy to be involved in this at the stage of you needing to convince UKRI that you've thought about what's going to happen to the data that you use and to implement it using cutting edge technologies like CRISPR detector, CRISPR Sherlock, CRISPR this, CRISPR that. Okay.
Yep, that's great. What we are expecting from you.
Good. Good.
Most of us is IT and AI expert. So our main role will be developing kind of the, let's say, model or prototyping to prove that our identification on TBC will be more efficient, cheaper, and faster, and more sensitive, as you mentioned before. And that's why we also have some of clinicians who also have expertise in healthcare. So anyway, thanks also for your recommendation. If anyone fit with our project, then we would like to welcome as well. So the more we have a collaboration, and it will increase our chance to win the proposal of, if not this year, we can try next year.
Yeah, sure. So
Sure.
I think it's really exciting what you're
Yeah.
doing, and it's clearly an important problem that you're trying to solve.
Yeah.
So that's why I'm delighted to be involved in any way you see fit for me to be involved with my expertise for diagnostics using CRISPR. There is another colleague here at Nottingham who does DNA sequencing. He works with Oxford Nanopore Technologies to do quite low cost, high resolution sequencing. His name's Matt Luce. He's a professor of DNA sequencing, really.
Yeah.
I don't know if he reads his emails or not, but I will contact him. I'll go and see him, and I'll discuss confidentially what you've just shown me here and see if he would also be interested, because if he were to be interested too as a expert consultant on your project or a named collaborator, or whatever phrase you want to use, then you would have both the diagnostic CRISPR expertise and the DNA sequencing expertise from the University of Nottingham, which might look persuasive to UKRI, because both myself and Professor Luce are known by UKRI.
Yeah. Okay, great. Thanks very much, Ed, for recommend us here. I think we can go through the proposal writing. So at the moment, we are still waiting from the government. What any question we have to fill up in the proposal. So I think yesterday, we just got the initial briefing from the government. There will be 70 applicants. I'm not sure how many will be taken from the proposal, but at the moment there are 70 across Indonesia. So I think it's quite competitive funding.
Yeah.
So, yeah. And so the next we will waiting from the government. What should we write up in the proposal? Anything, and we'll regularly update to you at. And if we need something like your information from your side, any kind of fill up or any concern that you have to fill, we'll send you, let you know. I know the time difference is quite quite significant. 7 hours there.
Yeah.
Yeah. And I think we are no problem in Indonesia because we used to watch APL, Manchester United, Chelsea, and etc.
Okay, good.
So don't worry if we have to wake up early in the morning or late in the night. So
You're used to it for watching the football.
yeah, for the APL, yeah.
Yeah. Good stuff.
Yeah,
Right, so look, I'm happy to carry on being involved. Keep me updated. We can always
yeah,
email
sure.
one another as well.
Yeah.
I will see what Matt Luce, my colleague, says about whether he wants to be involved. I'll update you on that, whether he says yes or no. I don't know. And we'll continue emailing, and then perhaps we can have another Zoom conversation
Yeah, sure, of
that
course.
suits you as this thing progresses. Yeah.
Yeah, great. Yeah. Okay, anyway, thanks for your time, Ed. I know you're also very busy, and we'll inform you later. Yeah.
Great.
Thanks
It's a
for
pleasure
calling.
to meet you all, and thank you for that presentation. It was really,
Yeah,
really
sure.
interesting stuff. Thank you. Bye.
Thank you. Thank you. Bye-bye, Ed. Yeah. Oke. Teman-teman, oke, kita udah dapat persetujuan nih dia mau join ya. Ya, dan dia rekomen satu lagi ahli DNA yang akan gabung juga di kita as expert ya. Selamat Pak Gede, udah di ACC. Tugas saya selesai ya.
Kelihatan Pak, nggak kelihatan Pak.
Nah, ini tangannya udah kelihatan. Nanti kan
Apanya Pak?
rekaman.
Oh rekamannya, iya. Sebentar ya Pak ya.
Oke,